Hello everyone and welcome to the new session of the biology lecture series. And today we'll be solving the NCERT Exemplar questions for the topic molecular basis of inheritance. So we have already solved the rest of the questions, which included the multiple choice questions, short answer questions and very short answer questions in the previous lectures. And today we'll focus on long answer type questions. So I would like to mention that before starting this lecture, that all the questions in this session are completely NCERT based. All right. So let's start with the first question. So the first question is, here we have to give an account of Hershey and Chase experiment and what did it conclusively prove. Okay. So Hershey's and Chase experiment. Okay. What did it prove? Now, if both DNA and protein contained phosphorus and sulfur, now do you think the result would have been the same? So, DNA, it is made up of what? DNA is made up of nitrogenous sugar, nitrogenous base. It is made up of pentose sugar and it is made up of a phosphate group. All right. Now, proteins, it consists of an amino group and a carboxylic group. All right, so this is clear. So this you have to keep in mind first. So here they are talking about phosphorus and sulfur. Okay, so now we have phosphorus. Phosphorus is present in where? DNA. All right. And sulfur. So sulfur is present in the protein. So there are some sulfur containing proteins. Okay, and they are made up of these sulfur containing amino acids. Now let's talk about the answer. Now Hershey and Chase, they conducted experiments on bacteriophage to prove that DNA is the genetic material. Alright, so this is the image. Here you can see this is the bacteriophage. What is a bacteriophage? Bacteriophage is a virus that can infect bacteria. So Hershey and Chase, they were working on E. coli. Okay, so this bacteria, this is representing E. coli. So the radioactive, first what happened? They labeled the bacteriophage with radioactive sulfur. Okay, so here you can see radioactive sulfur labeled bacteriophage. All right. Now, in the other set of experiment, what they did? They labeled the bacteriophage with the radioactive phosphorus. So you can see here, this is the radioactive phosphorus. Now, first let's talk about the structure of a bacteriophage. So bacteriophage, here you can see it consists of a head. Okay, it has a tadpole-like structure. First you have to remember, and here you can see this is the head. Okay, this is the head which is made up of proteins and it is known as the protein capsule or the protein coat. All right. And then you have this tentacles here. This is known as the neck region. Okay. Now, bacteriophage was labeled with radioactive sulfur, and in another set of experiment, bacteriophage was labeled with radioactive phosphorus. So now you know that DNA contains phosphorus and proteins contains the sulfur. So what they did in the next step, they infected the E. coli bacteria. So these are the E. coli bacteria with this bacteriophage. And then what they did they did the blending. Why they did the blending? To remove the viral code. So you know how a virus or a bacteriophage infects a bacteria. So it sits on the bacteria and then it transfers the genetic material inside the bacteria. Okay, and the protein capsule or the protein coat remains outside. Okay, so here you can see these are the protein coat. They remain outside and the genetic material is transferred inside. Okay, so blending was done to remove the viral coat, protein coat, to remove these viral protein coats. And then centrifugation was done. Why centrifugation was done? To separate the virus particles from the bacteria. Okay, and then in the end, what they noticed? They noticed that there was no radioactive sulfur detected within the cells. Okay, but radioactive sulfur were detected in the supernatant. So after centrifugation, what you get? You get the pellet and the supernatant. So here, the viral codes, they don't enter the E. coli or the bacteria. So they remain outside, and the supernatant consists of this viral coat. Okay. In supernatant, these viral coats are present. All right. So if the bacteriophage was initially labeled with the radioactive sulfur, so the protein coat will be labeled. So radioactive will be shown in the supernatant because supernatant consists of this protein capsid or the protein capsule. So radioactivity was seen in the supernatant. Whereas in this case, we use the radioactive phosphorus containing bacteriophage. So here you can see genetic material was transferred inside the cell, and the radioactivity was detected within the cells. And there was no radioactivity detected in the supernatant, which consisted of the protein coat. So here we were using the radioactive phosphorus. So radioactive phosphorus, okay, bound to the DNA, okay, because DNA consists of the phosphorus. All right, so that's why you can see DNA was transferred inside the cell, and DNA is the genetic material. Okay, so this experiment showed that DNA was the genetic material and protein was not the genetic material. The DNA from the bacteriophage was transferred, okay, inside the cell. All right. Now let's discuss about Hershey and Chase experiments again in detail. Now some bacteriophage virus were grown on a medium that contained radioactive phosphorus, okay, 32P, and some, and the another medium, some bacteriophage were grown in radioactive phosphorus containing media, and some bacteriophage are grown in another medium with radioactive sulfur, okay. Now, the virus is grown in the presence of radioactive phosphorus contained radioactive DNA. Okay, this you know now. Now, similarly, virus is grown in presence of radioactive sulfur. They will contain the radioactive proteins. Means the radioactive sulfur will be present in the protein coat. Okay. Now, both the radioactive virus types were then allowed to infect the E. coli bacteria separately. Then what happened? Now, soon after the infection, what they did? The bacterial cell was gently agitated in a blender. So, blending was done. Okay. Why was blending done? To remove the viral coats from the bacteria. Okay. Now, the culture was then centrifuged. And why was centrifugation done? To separate the viral particles from the bacterial cell. So, here is a difference here. Here was the viral coat. Now, centrifugation was done to remove the viral particles. Now, what was the observation and conclusions? Alright, now the only radioactive phosphorus was found to be associated with the bacterial cell. Okay, so the radioactive phosphorus was found to be associated with the bacterial cell, whereas the radioactive sulfur was only found in the surrounding media or in the supernatant and not inside the bacterial cell. Okay, so this shows that the radioactive sulfur was present in the protein coat and radioactive phosphorus were present in the DNA, and DNA was transferred inside the cell. Now, this indicates that only DNA and not protein coat entered the bacterial cell. Now, this proves that DNA is the genetic material which is passed from virus to the bacteria, and protein is not the genetic material. Okay, so this is clear. So, this was Hershey and Chase experiment. Now, the next part of this question was, if both DNA and proteins contain phosphorus and sulfur, the result, okay, might change. Okay, what will be the result? So, let's see case number 1. Okay, so if both radioactive phosphorus and sulfur was suppose present in the protein coat, okay, so suppose the proteins, okay, they are made up of both sulfur and proteins, or sulfur and phosphorus. So radioactive sulfur and radioactive phosphorus will be present in the protein coat. All right, then the radioactivity will be seen in the supernatant, because supernatant consisted of protein coat. Okay. So this will create a confusion whether protein was the genetic material or DNA was the genetic material. Okay. So now, because of the presence of both sulfur and phosphorus in DNA or in protein, so it will be very difficult to decipher whether genetic material is DNA or RNA. Okay. So in case 2, let's see, if both radioactive sulfur and radioactive phosphorus were present in the DNA, then what will happen? Radioactivity will be detected in the cells or within the cells. So this will create a confusion here. All right. This is clear. Now, next question. That during the course of evolution, why DNA was chosen over RNA? So, initially RNA was the genetic material. Okay. But later on, now you can see DNA is the genetic material. Okay. Now, RNA initially they played a lot of roles in a lot of things. It acts as a catalyst. It has enzymatic activity. Okay. It has some role in the metabolism. Okay. It has some roles in the chromosomal structure and all. Okay. It also had role in splicing also. Okay. So that's why RNA was the genetic material. But RNA was very unstable. Okay. Why it is unstable? Because of the presence of 3' hydroxyl group. Whereas in DNA, 2' hydroxyl group. Whereas in DNA, 2' hydroxyl group is absent. Okay. So DNA consists of deoxyribose sugar, whereas RNA consists of ribose sugar. Okay. So DNA lacks a OH group, whereas RNA, it has the OH group on the second position. Now you have to give reasons by discussing the desired criteria in a molecule that can act as a genetic material. So you have to describe, okay, some criterias, okay, for a molecule to be called as a genetic material, and how you have to describe it in the light of biochemical differences between DNA and RNA. Okay. Now, a molecule can act as a genetic material. That act as a genetic material must fulfill the following points. Okay. So these are the criteria. It should be able to generate its replica. Means it should be able to replicate. Okay. Replication should be there. So as to produce more number of copies. So this is important for the survival of the organism. And what is the basis of replication? Basis of replication is the complementary base pairing. Okay. So A pairs with T and G pairs with C. Alright. So this is the basis of DNA replication. Second criteria, it should chemically and structurally be stable. Okay. So RNA was unstable. So that's why slowly DNA become the genetic material now, because DNA is quite stable. Alright. DNA is chemically and structurally both stable. And RNA, okay, I've already told you, act as a catalyst. It has enzymatic activity, and that's a reason it is chemically unstable. Alright. Now, third criteria, it should provide the scope for slow changes that are required for evolution. Okay. So, mutation should happen, that too at very slow rate. Okay. So, that material or that molecule should show a limited mutation, that too at a very slow pace. All right. And why mutations are required? Mutation required for evolution. Okay. So, environment is changing. So, you should allow some mutation to adapt to the environment. Now, fourth criteria, it should be able to express itself in the form of Mendelian character. So, it should follow Mendelian inheritance. Okay. So, these are the four criteria on which a molecule can be called as the genetic material. Now, there are several reasons why DNA is considered a superior genetic material. And we will talk about this in the terms of biochemical differences. Now, DNA chemical stability, okay, is enhanced by the presence of the 2' H. So, I've already told you, DNA consists of deoxyribose. Okay. So, at the second position of the pentose sugar in this, which is deoxyribose, instead of OH, H is present. Okay. So DNA's chemical stability is enhanced by the presence of 2' H in the deoxyribose sugar, making it more resistant to chemical reactions compared to RNA. Okay, and RNA, it contains chemically reactive 2' OH, which is present in the ribose sugar, which makes it unstable. Now, RNA also has the catalytic properties and is very reactive, while DNA lacks catalytic properties and is very less reactive. Okay, now third point, thymine. DNA is made up of 4 nitrogenous bases. It consists of 4 nitrogen bases: adenine, guanine, thymine, and cytosine. So thymine is present in the DNA and it is less prone to tautomerism. So tautomerism causes mutations. Alright. Whereas RNA consists of uracil, which is more prone to tautomerism and mutations. So this is also a reason why RNA is unstable. Okay. And because of these reasons, DNA evolved as a genetic material which is most chemically and structurally stable. Now let's talk about next question. Okay. Now here we have to give an account of post-transcriptional modifications of a eukaryotic mRNA. Okay. This is clear? So you know about the central dogma. Okay. DNA transforms into RNA and this process is known as transcription. And then RNA, okay, then is translated into proteins. Okay. And this process is known as translation. So here they are talking about post-transcriptional modification. So what happens after the transcription? Okay, what are the modifications that are happening after transcription? Means after the formation of the primary RNA. Okay, also it is also known as the pre-mRNA. All right, or it is also known as hnRNA, heterogeneous nuclear RNA. All right, now here I've given you a thinking process. This will help you understand this question. Now the post-translation modifications, they include the modification of the mRNA transcript that is synthesized by the RNA polymerase II. Okay. And here I've written in eukaryotes. So the post-transcriptional modification, it takes place in eukaryotes. Okay. And prokaryotes, they lack post-transcriptional modification. Okay. So RNA polymerase II, it synthesizes, okay, primary transcript or primary mRNA. All right. And then it is modified. So let's talk about now post-transcriptional modification. So the primary transcript which is produced, which is known as the hnRNA, okay, heterogeneous nuclear RNA, okay, also known as the primary transcript or the pre-mRNA. Okay, so the primary transcript which are produced initially, they are non-functional, okay, and they contain both the coding regions are the exons, and they also consist of the non-coding regions which are known as the introns. Okay, so both of these exons and exons are present in the primary transcript, and primary transcript is also known as heterogeneous nuclear, so H for heterogeneous and then nuclear RNA. Okay, this is clear. Now in eukaryotes we require only exons because exons are the coding regions, means they code for a particular polypeptide. Okay, introns, they are non-coding, so they have to be removed. All right, and they are removed by a process known as intron splicing, which is one of the post-transcriptional modification. All right, so now in eukaryotes, 3 types of RNA polymerases are found. Okay, let's first discuss about this. Okay, 3 types of RNA polymerases are found in the nucleus. First one is RNA polymerase 1. Okay, which transcribed what? Which transcribed ribosomal RNAs. Which kind of ribosomal RNA? 28S and 5.8S ribosomal RNA. Okay, so RNA polymerase 1, they transcribe 28S and 5.8S ribosomal RNA. Okay, or you can write this in bracket. Okay, if you are writing like this. Now RNA polymerase 2, okay, it transcribes the precursor of the mRNA known as the heterogeneous nuclear RNA or hnRNA. Okay, this we have already discussed, and this hnRNA consists of both introns and exons. Now RNA polymerase 3, it transcribed the transfer RNA, 5S ribosomal RNA, okay, and snRNA. What are snRNA? They are small nuclear RNAs. So this is the function of RNA polymerase 3. So you can see here RNA, ribosomal RNA are of different type, 28S, then 5.8S, and 5S. Okay. Now, let's again come back to the post-translational modification. Now the hnRNA, okay, also known as the primary transcript, it undergoes two additional processes. So these processes are known as post-transcriptional modification. And these two additional processes are known as, the first one is capping. So you can write this is the first one, tailing is the second one. So a 5' guanine cap is added, and tailing means addition of the adenine residues. Okay. Let's talk about this. So in capping, what happens? An unusual nucleotide known as methyl guanosine triphosphate. This is added where? To the 5' end of the hnRNA. So this is known as capping, okay, which takes place at the 5' end. So this capping has a role. Okay, we'll talk about this. So, okay, we'll talk about this when we will study the translation. Okay, the small subunit of the ribosome, it recognizes this 5' guanosine. Okay, this 5' methyl guanosine triphosphate, and then it binds there. Okay. Now, in tailing, what happens? Adenylate residues, okay, around 200 to 300, they are added at the 3' end in a template independent manner. Okay. So 5' cap and 3' tail, 3' adenyl tail is added. So these are the two processes. Okay. Now, apart from these two processes, there is one more process known as splicing. So splicing is the third process. Okay. Now, what happens here? The hnRNA undergoes the process where the introns are removed. So introns are the non-coding regions, so they have to be removed, and exons are then joined together to form the mature mRNA. And this process is known as splicing. Okay, this is clear. So these are the three post-translation modification that is happening in eukaryotes. So we are talking about eukaryotes, okay, and there is no post-transcription modification in prokaryotes. So in prokaryotes, what happens? There is no defined nucleus, okay, there is no nuclear membrane, there is no nucleus, and the genetic materials lie within these cells. Okay, so what happens there? Transcription and translation, it is happening simultaneously. All right, and both transcription and translation, it happens in the cytoplasm. Whereas in eukaryote, what happens? We have a defined nucleus, we have true nucleus, and transcription takes place within the nucleus, and then the primary transcript is produced, and then it is processed, and a mature RNA is produced, and then this mature RNA is transferred from the nucleus into the cytoplasm where the translation takes place. Okay, so this is the difference between prokaryote and eukaryotic transcription and translation. So here I've written a note here. In prokaryotes, mRNA does not require any processing because all the processes are happening in the in the cytoplasm. Okay. Now, this is the figure here showing you the post-translational modification. So initially here you can see the RNA synthesis is happening. Okay. Initially, hnRNA is produced. And what happens? First, capping takes place. Okay. So capping you can see here. So here at the 5' end, this methyl guanosine triphosphate, you can see there are 3 phosphate here. This is added to the 5' end. All right. And then you can see here, we have the exons and introns are present. All right. So the primary transcript consists of both exons and the introns, and then by the process of RNA splicing, all these introns are removed. So these introns are removed and the exons are joined together. And then what happens? Polyadenylation. Okay, first step is capping, second is splicing, and then polyadenylation, where a poly-A tail is added or adenylate residue, they are added at the 3' end. And here you can see this is the mature mRNA with 5', okay, 5' cap and 3' poly-A tail, and here introns have been removed. So, okay, this is clear. Now, let's talk about the next question. So, this is again a very straightforward question. Here you have to discuss the process of translation in detail. Okay, now translation, you know, is the conversion of RNA into polypeptides or proteins. So, this process is known as translation. Now, translation is the process of synthesis of protein from what? From the mRNA with the help of ribosomes. So, ribosomes are the site of protein synthesis. A translational unit in the mRNA, okay, from 5' to 3' end, it comprises of a start codon. Okay, so for the initiation of the translation, what is required? Start codon, which is usually AUG, and it codes for methionine. Okay, so start codon, a region coding for a polypeptide we require. This is the second requirement. And then in the end, to terminate the translation process, we require stop codons. All right, now there are 3 types of stop codons. We will talk about this. And apart from this, untranslated regions at both 5' and 3' end are required. So these untranslated regions are also very important. So they help to maintain the stability of this mRNA. Okay, they also help in the subcellular localization of this RNA, and it also helps in the transport of the mRNA from the nucleus to the cytoplasm. So this is clear. Now there are 3 stages of protein synthesis. First is the activation of the amino acid. Then second is the charging of the transfer RNA, and third is the polypeptide chain formation. Okay, so initially, activation of the amino acid is required. Okay, and then charging of the transfer RNA, and third is the protein chain formation. Okay, now first let's talk about the activation of amino acid. So this is a very crucial step. Without this, the protein formation cannot begin. Okay. Now, activation of amino acid, what happens? The amino acyl transfer RNA synthesis. So these are the class of enzymes. Okay. Amino acyl transfer RNA synthesis. There are 2 types, class 1 and class 2. So these are the enzymes that facilitate the coupling process by linking each amino acid to its corresponding transfer RNA. So you know transfer RNA, it consists of an amino acid receptor site. Okay. So these enzymes, they link each amino acid with its corresponding transfer RNA. So transfer RNA consists of one, I have told you, amino acid receptor site. It consists of the anticodon loop, where the anticodon is present and it recognizes the codon which is present in the messenger RNA. It also consists of D loop and the T loop. Okay. Now what is happening in activation of amino acid? First is the transfer of amino acid to the transfer RNA. So here what happens? The carboxylic group of the amino acid forms a covalent bond with the alpha phosphate of the ATP molecule. Okay, and this results in the release of inorganic pyrophosphate, which is known as which is denoted like this, PPI, and there is a formation of a 5' amino acyl adenyl intermediate, which is known as AAAMP. Okay, so this is amino acyl adenylate intermediate. So this is the reaction which happens. Amino acid, okay, in the presence of ATP, it forms amino acyl adenylate intermediate, and a pyrophosphate is released. Okay, now Then what happens? Subsequently, a nucleophilic attack on the amino acyl adenylate intermediate means AA-AMP. Okay, there is a nucleophilic attack on AA-AMP which leads to the binding of an amino acyl group to the 3' OH of the transfer RNA. Okay, and this is accompanied by the release of AMP molecule, adenosine monophosphate. And this is the reaction. So amino acyl adenylate intermediate was formed in the previous step. This now attaches with the 3' OH of the transfer RNA. And what happens? Amino acid, okay, now get attached to the transfer RNA and AMP is released in this process. So here you can see just amino acid is transferred to the transfer RNA and AMP is released. So this is the activation of the transfer RNA or the activation of the amino acid. Now, third step I've already told you was the protein synthesis. Now the actual protein synthesis takes place. So this was pre-protein synthesis, activation of amino acid all. Now this is the actual steps that are involved in the protein synthesis. So it comprises of 3 sequential steps. First is initiation, second is elongation, and third is termination. Okay, now first let's talk about the initiation step. So here what happens first? The cap region of the mRNA. What was the cap region? The 5'. Okay, the 5' cap region of the messenger RNA. It binds to the smaller subunit of the ribosome. Okay, so ribosome we know it consists of 2 subunits, larger subunit and the smaller subunit. Alright, so the smaller subunit binds to the 5' cap region of the messenger RNA. And then what happens? You know about the large ribosomal subunit. The large ribosomal subunit, it consists of 2 sites, A site and the P site. Okay, so now initially smaller subunit of the ribosome come and it attached at the 5' cap region of the mRNA. And then what happens? The smaller subunit, okay, smaller ribosomal subunit, it first bind to the initial transfer RNA. So what is happening here? First, smaller subunit binds to the cap region and then initiation transfer RNA comes, okay, which has the amino acid. So this initiation transfer RNA then binds to the smaller subunit, okay, and then the larger subunit comes, okay, so that the initiation codon AUG lies at the P site. So initially what happens? The initiation codon, which is the AUG, it lies at the P site. So what is happening here? First, the smaller subunit of the ribosome, it attaches at the 5' cap region of the mRNA, and then the initiation transfer RNA binds to this region, okay, and then what happens in the end? Larger subunit, it attaches to this entire complex and the initiation complex is formed. So initiation complex consists of what? It consists of smaller subunit of the ribosome, then it consists of the initiation transfer RNA, and it consists of what? The larger subunit of ribosome also. Okay, so it is made up of 3 things: smaller subunit of ribosome, larger subunit of ribosome, and initiation transfer RNA. And this complex is arranged in such a way that the initiation codon, okay, lies in the P site. Okay, I've already told you the ribosomal subunit, larger one, consists of A site and the P site. So initially AUG is present in the P site. Now the initiation transfer RNA, okay, this one, initiation transfer RNA, okay, it consists of methionine transfer RNA. So initiation transfer RNA is also known as methionine transfer RNA that binds to the P site where the initiation codon is present. Okay. Now initiation codon consists of AUG. Okay. So AUG codes for methionine. So that's why transfer RNA will bring methionine and it is known as methionine transfer RNA. Now next step is elongation. Now in elongation what happens? Another charged amino acyl transfer RNA complex binds to the A site. Okay, so initially the first transfer RNA, which is known as the methionine transfer RNA, that will attach itself to the P site because the initiation codon is present in the P site. Now the second, okay, charged amino-acyl transfer RNA means the second transfer RNA, you can say, which is carrying the second amino acid along with it, it binds to the A site of the ribosome. Okay. Now what happens? Now peptide bond formation takes place. Okay, so this is also comes under elongation only. Now a peptide bond is formed between the carboxyl group, means COH, of the amino acid at the P site. Okay, and there is one more amino group, okay, which is present at the A site. Okay, so a peptide bond is formed between the COH of the P site and the amino group, which is the NH group, okay, which is present at the A site. Now we have two transfer RNA, two charged transfer RNA. One was at the P site, another was at the A site. So now there is a peptide bond formation between the two amino acid. Okay, and this reaction, peptide bond formation, is catalyzed by the enzyme peptidyl transferase. Okay, now what happens? Now the ribosome slides over the mRNA. So what happens? Now the ribosome will slide over, or you can say move over the RNA from codon to codon. In which direction? 5' to 3' direction. And according to the sequence of the codons, amino acids are attached to one another by peptide bonds and a polypeptide chain is formed. So the ribosome is moving over the mRNA or sliding over the mRNA, okay, from codon to codon. Okay, codon, you know, is made up of 3 nitrogenous bases. Okay, so and according to the sequence of the codons, corresponding amino acid will be attached to the transfer RNA, and then peptide bond will form between the amino acid present in the P site and the A site, and then polypeptide chain formation will occur. Now third step is termination. So what happens in termination? Now when the A site of ribosome reaches the termination codon. Okay, so here some codons are required for termination. So these are known as termination codons. Termination codons are also known as stop codons or nonsense codons. And why they are called as such? Because they don't code for any amino acids. Okay. Now, when the A site of the ribosome reaches the termination codon, which does not code for any amino acid, no charged transfer RNA binds to the A site. Alright, this is clear. So what happens? Now the dissociation of polypeptide from the ribosome takes place. Okay, and this is catalyzed by the release factor. So here, first, they will encounter the stop codons. Okay, and after they have encountered the stop codons, and then release factor will come and they will help in the dissociation of the polypeptide from the ribosomes. Okay. Now there are 3 termination codons. I've already told you. Okay. One is UGA, second is UAG, and third is UAA. Okay. These are clear. And termination codons are also known as stop codons or nonsense codons. So whenever the transfer RNA, which was present in the A site, they come across these termination codons, then what will happen? Termination will stop and release factor will come and they will dissociate the polypeptide from the ribosome. Okay. Now this is the process showing you the process of image, showing you the process of translation. So I've shown you translation. First was the activation of the amino acid. Okay. So this was pre-protein synthesis. Okay. And then protein synthesis consisted of these steps: initiation, elongation, and termination. So here you can see, here this is the messenger RNA. Okay. Here you can see this is the transfer RNA which is bringing the corresponding amino acid. This is the AUG, initiation codon. Okay. Here you can see initiation or the first codon which consists of AUG. And the transfer RNA is consists of the anti-codon loop. Okay, so AUG was the codon, initiation codon. So anti-codon of it will be UAC. Okay, and AUG codes for methionine. So here it will bring methionine. Alright, so here I've told you at the 5' region, small ribosomal subunit will attach. All right. And then the initiation, this is the initiation transfer RNA will come, and then the large subunit of the ribosomal will then attach. And then large subunit of the ribosome, it consists of the A site and the P site. And this complex is arranged in such a way that the AUG initiation codon, so this is the initiation codon, this is present at the P site. Okay, and this entire structure is known as initiation complex, and it is arranged in such a way that the initiation codon AUG lies at the P site. Okay, and again I would like to mention that the large subunit, this is the large subunit. Okay, this is the small subunit. So L for large, this is small subunit, and this one is the initiation transfer RNA. All right, this is clear. And then second step was elongation. So here another charge transfer RNA will come and it will attach at the A site. Okay, and this is the second codon. Okay, and correspondingly it will bring the amino acid. Now what will happen? The ribosome will slide over the mRNA. Okay, and what will happen as it slides? Okay, this transfer RNA which were present in the P site will then enter in the E site. E site means the exit site, and then here you can see here is the exit of the first transfer RNA, first initiation transfer RNA which was carrying methionine. All right, and then as it moves along the messenger RNA, you can see another transfer RNA. So this is the third activated transfer RNA that will come at the A site. Okay, so the ribosome will slide or the move over the messenger RNA. Okay, and the transfer RNA which were present in the A site will move to the P site. Okay, and the transfer RNA which were present in the P site will move to the E site, which is known as the exit site. Okay, now what happens in the termination? I've already told you, when the ribosome encounter the stop codons or the terminator codons, then what will happen? Release factor will come, and what will happen? It will help dissociate the polypeptide from the ribosome. So here you can see there is a dissociation of the initiation complex and the polypeptide is released. So this happens during termination. Okay, so this is clear. Again, let's summarize this. So initially, what happens during initiation? There is the small subunit. So this is the small ribosomal subunit. Okay, it attaches to the 5' guanine, and then what happens? Next stage, initiation codon will come. Then what happens? The large subunit of the ribosomal will attach, and then the initiation complex is completed. Okay, and initiation complex is arranged in such a way that the AUG is present in the P site first. Okay, then what happens? So this is the P site here. This is the A site here. Next transfer RNA will then, okay, appear or it will attach to the A site. So this is the A site, this is the P site. Next transfer RNA will attach itself to the A site here. I am writing here. This is the A site, this is the P site, this is the E site, exit site. And then the transfer RNA will, and then the ribosome will move along the messenger RNA, and then the transfer RNA present in the P site will then enter the E site, and then it will exit. E site means exit. All right, and the next amino acid, all right, can then bind to the A site, and then peptide bond formation will take place during elongation. And then what will happen during termination? When the ribosomes they encounter the stop codons, then termination of the translation will take place and release factor will come. Okay, so these are the release factors, and they will help in the dissociation of the polypeptide from the ribosome. Now, next question. Here we have to define an operon giving an example. Okay, and then you have to explain an inducible operon. So let's talk about this. The concept of operon was first proposed in 1961 by Jacob and Monod. Okay, now what is an operon? An operon is a unit of prokaryotic gene expression which includes coordinately regulatory structural genes, and it also consists of the control elements which are recognized by a regulatory gene product. Okay, so lac operon is a polycystonic gene. It is present in the bacteria, prokaryotes. Now, what are the components of the operon? First is the structural gene. What is the structural gene? It is the fragment of DNA that transcribe mRNA for the polypeptide synthesis. So, structural genes, okay, so these forms the polypeptide or the protein. So, they are, okay, transcribed first into mRNA, and from mRNA, they are transcribed into the polypeptide. Now, it also consists of a promoter region. So, what is the promoter? It is a sequence of DNA where RNA polymerase binds and initiate the transcription of the structural genes. So, this is known as promoter. And what is operator? Sequence of DNA adjacent to the promoter. So, operator is present adjacent to the promoter where specific repressor protein binds. Alright, so this is known as operator. So, repressor protein binds at the operator. This you have to remember. And it is present adjacent to the promoter. Okay. Now, the operon also consists of this regulator gene, or you can call it as regulatory gene. Alright. Now, this gene codes for the repressor protein. So, repressor protein, I've already told you, it binds to the operator. Okay. So, genes that code for the repressor protein that binds to the operator and suppresses its activity, as a result of which transcription will be switched off. So, regulatory gene, okay, it codes for a repressor protein, and this repressor protein binds to the operator, and when it binds to the operator, then the lac operon will be switched off, or you can say the transcription is switched off. There is no formation of the polypeptides or the proteins. Now, there is an inducer which is also required in the lac operon. So, we are talking about the inducible lac operon. So, here lactose is the inducer. Okay, and what is an inducer? It is the substrate that prevents the repressor from binding to the operator. Okay, so you know repressor binds to the operator, but in the presence of the inducer, repressor is not able to bind to the operator. So, this is known as the inducer. It is a substrate such as the lactose. Okay. Now, in the presence of the inducer, the repressor cannot bind to the operator. Okay. And as a result, transcription is switched on. Alright. Now, the inducer, it is chemically very diverse, and it can be a metabolite or a hormone. Now, let's talk about the inducible operon system. So, an inducible operon system is a regulatory unit of genetic material which is switched on in response to the presence of a chemical, for example, lactose or lac operon of the E. coli. Alright, so what is an example of inducible operon system? Lac operon of E. coli, where inducer is a lactose. So, lactose acts as an inducer in lac operon of E. coli. Okay, this is clear. Now, let's talk about this lactose operon. Okay. Now, lactose operon, it consists of these genes, LAC-Z, Z, Y, and A gene. So, these are the three structural genes which are present in the lactose operon. So, these genes are transcribed from a lac transcription unit under the control of a single promoter. So, what is the unique feature of the polycystonic gene? Or you can say the lac operon. So, lac operon is an example of a polycystonic mRNA or polycystonic gene, you can say. Okay. So, what happens in lactose operon or polycystonic genes or polycystonic mRNA? The structural genes, okay, these genes, they are under the control of a single promoter. So, these happens in prokaryotes. Alright. Now, they encode enzymes required for the use of lactose as a carbon source. So, lac operon is involved in the metabolism of the lactose. Now, apart from the structural genes, it also consists of this regulatory gene which is denoted by I. Okay. So, the LAC-I gene product, the LAC repressor product. Okay. So, LAC-I, known as a regulatory gene, okay, it produces repressor. Okay, the LAC-I gene product is known as the LAC repressor. Okay, this is expressed from a separate transcription unit upstream from the promoter. So this LAC-I gene or the regulatory gene is present upstream from the operator, means on the left side you can say. Now the LAC operon consists of three structural genes. This we have already discussed: Z gene, Y gene, and the A gene. It also consists of the operator, promoter, and the separate regulatory gene, which is denoted by I. Now the three structural genes, they transcribe the polycistronic mRNA. This I've already told you. So here you can see in the absence of the inducer. So this is a LAC operon first of all. Okay, this is a LAC operon. It consists of a promoter. This is the regulatory gene I. This is again a promoter here. This is the operator. These are the structural genes Z, Y, and A. These are the structural genes. So what is happening here in the absence of the inducer? Okay, inducer means lactose. In the absence of the lactose, what is happening? This regulatory gene, this produces a repressor mRNA, and then our LAC repressor is produced. Now, in the absence of the inducer, repressor binds to the operator. All right, and then what happens? Repressor binds to the operator region and prevents RNA polymerase from transcribing the operon. But what happens in the presence of the inducer? Now, inducer is present, means lactose is present. Then this, here you can see, first of all, the regulatory gene, it produces the repressor. So this is the repressor. Now what happens in the presence of the inducer? This inducer binds to this repressor and inducer repressor complex is formed first, and because of this binding, the repressor becomes inactive. Now this repressor cannot bind to the operator. Okay, it cannot bind to the operator because it is inactive now. And why it is inactive? Because inducer has bound here to the repressor molecule. Okay, and now what happens in the presence of inducer? Now the transcription can take place. Okay, here LAC mRNA is produced and then polypeptide or proteins are produced. So LAC-Z gene codes for galactosidase. Okay, LAC-Y gene, it codes for permease, and LAC-A gene, it codes for trans-acetylase enzyme. And these enzymes are, or these proteins are involved in lactose metabolism. So here, the gene Z, it codes for beta-galactosidase or BGAL enzyme, which breaks lactose into galactose and fructose. Okay. Now, if you talk about gene Y, it codes for permease, which increases the permeability of the cell to the lactose. Okay. Now, LAC-A gene or gene A, sorry. Okay. So this was gene Z, this was gene Y. Gene A codes for the enzyme trans-acetylase, which catalyzes the trans-acetylation of lactose in its inactive form. So here you can see here, gene Z, it codes for beta-galactosidase and it breaks lactose into galactose and glucose. Gene Y, it codes for permease, which increases the permeability of the cell to lactose. So if permease is not produced, lactose will not be able to get inside the cell. Okay, and gene A is code for the enzyme trans-acetylase. Now, when lactose is absent, this I've already discussed. When lactose is absent, the regulatory gene, okay, produces the repressor mRNA, which translates into repressor. So repressor protein is produced. Now, this repressor protein, it binds to the operator region of the operon, and as a result, prevents the RNA polymerase to bind to the operon. Okay, so this means the operon is switched off. Now, in the presence of lactose, what happens? The lactose acts as an inducer, which binds to the repressor protein and forms an inactive repressor. Okay. Now, the repressor fails to bind to the operator region now. And what happens? The RNA polymerase now can bind to the operator, okay, and transcript the LAC mRNA. Means the LAC operon is switched on. Okay. Now, we know that the LAC mRNA is polycistronic. We already know that it produces all the three enzymes, beta-galactosidase, permease, and trans-acetylase. So here you can see the structural genes, they are under the control of a single promoter. And these kind of operon, they are known as the polycistronic mRNA. Okay. Now, the LAC operon is switched on. Okay, and all these three enzymes are being produced in the presence of the lactose. That's why it is known as inducible system. The next question is, there is a paternity dispute for a child. Which technique can solve the problem? And we have to discuss the principle involved. Now, DNA fingerprinting is a technique used in solving the paternity dispute for a child. Now, DNA fingerprinting technique of determining the nucleotide sequences of certain areas of DNA which are unique to the each individual. Okay, so DNA fingerprinting, what does it include? It is a technique of determining the nucleotide sequences. Okay, of determining the nucleotide sequences of, you can say, certain areas of DNA. Okay, not all the areas, but certain areas of DNA which are unique to each individual. Okay, now the basis of DNA fingerprinting. What are the bases of DNA fingerprinting? It is DNA polymorphism, or you can say VNTR. We'll talk about this VNTR. What is DNA polymorphism? DNA polymorphism means presence of two or more variants of a DNA sequence. Okay, presence of two or more variants of a specific DNA sequence. This is known as DNA polymorphism. Now, although the DNA from different individuals is more alike than different, there are many regions of the human chromosome that exhibit a great deal of diversity. So what does this mean? This means that although the 99.9% of the human genome is similar, but there are some regions that exhibit a great deal of diversity. Okay, now such variable sequences, they are termed as polymorphic. Polymorphic, I've already told you, means presence of many forms. So this is known as, so in case of DNA, if we talk about, it is known as DNA polymorphism, which means presence of many forms of a specific sequence of DNA. Now, a special type of polymorphism known as VNTR. So VNTR is the basis of DNA fingerprinting. Okay, so it is a type of DNA polymorphism only. What is the full form of VNTR? Variable number of tandem repeats. So this is composed of repeated copies of DNA sequence that lie adjacent to one another on the chromosomes. So what are VNTR? So VNTR, they are nothing but they are composed of the repeated copies of a DNA sequence. So each individual, they differ in the copy number of these repeated sequences. Okay, so copy number is unique to an individual. Alright, now since polymorphism is the basis of genetic mapping of humans, okay, so this can also be used in the pattern to solve the paternity disputes. Now coming to next question. So this question is, give an account of the methods used in sequencing the human genome. Alright, now let's talk about this. The sequencing of human genome has made it possible to understand the link between various genes and their functions. Okay, so human genome project was started. It was a collaborative effect, and after the human genome project, now we know that, okay, we have around 25,000 to 30,000 genes. Okay, and now we know the functions of some of the genes also, but the functions of over 50% of genes is still unknown. Okay, but for some genes we know the functions. All right, and we can use this information to do the pedigree analysis, to treat diseases, okay, genetic diseases, or to trace the inheritance patterns of the genetic diseases. All right, so if there are any gene defects that express as disorders or that increase the susceptibility of an individual to a disease, then specific gene therapies can be worked out. Okay, so we can apply this information. Okay, we can use the concept of, okay, or the process of gene therapies. Okay, if we know about the genome of a human, or if we have the information about the genome of the organism. Now, what are the methodologies of human genome sequencing? Okay, now the following methods can be used. Now, some of the methods, or I have given you two major approaches for human genome sequencing. So first is the express sequence tag, also known as ESTs. So in this method, what happens? So this method focusing on identifying all the genes that are expressed as RNA. Okay, that's why the name given expressed. Okay, so all the genes, okay, that can be expressed as RNA. Okay, so you know some of the nucleotide sequences, they cannot be expressed into RNAs. They are not coding. Okay, they are non-coding. So express sequence tag is focuses on identifying all the genes that can be expressed as RNA. Now, second method is sequence annotation. Okay, so in this what happens? So in this approach, or it is an approach of simply sequencing the whole genome. So what they do in sequence annotation, that's why the name sequence annotation, they sequence the whole genome first. Okay, that contains all the coding and the non-coding sequences. So in express sequence tag, we were only focusing on the coding sequences which can be expressed. Here we have sequenced the entire genome, and you know genome consists of both the coding and the non-coding sequences. Okay, and then what they do, and then later you assign different regions in the sequence with its function. Okay, so what is happening here? You sequence the whole genome which contains both the coding and the non-coding sequence, and then what you do, then you assign the functions to different regions of the sequence. Okay, now later you tell, okay, this region has this function, this region has this function. Later on, you do this after the entire sequencing is done. So this is the difference between sequence annotation and expressed sequence tags. Alright, this is clear. Now, for sequencing, what you do? First, you isolate the total DNA from the cells and you break down them into relatively small size. Okay, so you break down the DNA into fragments, and then what you do? These DNA fragments are then cloned in suitable host using suitable vectors. Now, when bacteria is used as a vector, they are called the bacterial artificial chromosomes, and when yeast is used as a vector, they are called yeast artificial chromosomes. Okay, so this is clear. Now, Frederick Sanger, they developed the principle according to which the fragments of the DNA are sequenced by automated DNA sequences. Now, on the basis of overlapping regions on the DNA fragment, these sequences are arranged accordingly. Okay, and for alignment of these sequences, specialized computer-based programs were developed. This is clear. Now, finally, the genetic and physical maps of the genome were constructed by collecting information about certain repetitive DNA sequences and the DNA polymorphism. Okay, and these were based on endonuclease recognition sites. Okay, this is clear. The genetic and the physical maps of the genome were constructed by collecting information about the repetitive DNA sequences. Okay, and the DNA polymorphism. Okay, this is clear. Now, next question. Here you have to list the various markers that are used in DNA fingerprinting. Alright, this is clear. Now, let's talk about the answer here. Now, Dr. Alex Jeffrey, he developed the technique of DNA fingerprinting in an attempt to identify DNA marker for inherited diseases. Now, DNA fingerprinting uses short nucleotide repeats called variable number of tandem repeats. This already we have discussed. The basis of DNA fingerprinting is VNTR. So, DNA fingerprinting uses this VNTR as markers, and VNTR vary from person to person and are inherited from one generation to the next generation. And only closely related individuals, they have the similar VNTRs. Okay, so I've already told you each individual, okay, they differ in the copy number of this VNTRs. All right, and on this basis, DNA fingerprinting is done. And if the individuals are closely related, then they have similar pattern of this VNTR. All right, this is clear. So let's start with the next question. So in the question, it is saying that replication was allowed to take place in the presence of radioactive deoxynucleotides, precursors in the E. coli that was mutant for DNA ligase. Okay, so this E. coli bacteria, this is mutant for DNA ligase, means there is no DNA ligase activity. And if there is no DNA ligase activity, the DNA fragments, they won't be able to join together. Because what is the function of DNA ligase? It joins together the two DNA fragments with the help of 3' to 5' phosphodiester bond. So a bond is formed between the 3' OH and the 5' phosphate of the nucleotide, and this is known as 3' to 5' phosphodiester bond, which is formed by DNA ligase. Now this E. coli is mutant for DNA ligase, so the fragments known as the Okazaki fragments that are formed on the lagging strand during the replication, they won't be able to join together. Okay, so in replication there is one leading strand. All right, on that leading strand the DNA synthesis is continuous, whereas on the lagging strand the DNA synthesis is discontinuous, and Okazaki fragments are formed which are joined together later by the DNA ligase. Now coming back to the question, now here newly synthesized radioactive DNA, it was purified later and the strands were separated by denaturation. Okay, now then it was centrifuged using the density gradient centrifugation. Now you have to tell which one of the following is correct. So option number A, so this graph, here you can see there are two peaks. One is high molecular weight peak, another one is low molecular weight peak. Okay, so here you can see two peaks here. One is high molecular weight, one is low molecular weight. Now you can see graph number B. Okay, here a high molecular peak is formed and there is no low molecular weight peak. Okay, now let's talk about option number C. So here you can see, here is the radioactivity. Here there is no high molecular weight peak and there is no low molecular weight peak present, but there is an intermediate peak present. All right, now let's talk about option D. In option D, you can see only low molecular peak is observed and there is no high molecular peak. All right, now let's talk about the answer here. Now, in this case, you can see E. coli, which is mutant for DNA ligase. All right, in this case, there will be no further joining of the Okazaki fragments that are produced on the lagging strand. Okay, this is clear. So what is happening here? So in the second generation, what will happen? They will produce low molecular weight fragments, okay, that are unable to undergo additional replication. Okay, so Okazaki fragments won't be able to join together because the organism E. coli is mutant for DNA ligase. So a low molecular weight fragments will be produced that are composed of the Okazaki fragments and they are unable to undergo additional replication. So the leading strand will undergo replication in each subsequent generation. Okay, because in leading strand there is continuous synthesis of DNA. Here there is continuous synthesis in 5' to 3' direction. Continuous synthesis of DNA. In which direction? In 5' to 3' direction. So you have to remember DNA replication is always in 5' to 3' direction. That's why on the lagging strand, the DNA fragments are produced in fragments. Okay. Now, what happens? So as a result, the two peaks will emerge. The leading strand's newly synthesized continuous DNA fragment, it will exhibit a peak indicating a high molecular weight. Okay, and the Okazaki fragments which are produced on the lagging strand, it will display a peak reflecting low molecular weight. Alright, this is clear. Now, what will happen? Ultimately, it will result into the formation of both high molecular weight fragments, okay, on leading strand and low molecular weight fragment on the lagging strand. Hence, only graph A could be the appropriate result for the for the appropriate results after the centrifugation. So there will be two peaks, one of the high molecular weight, one of the low molecular weight. So high molecular weight peak represents what? It represents the DNA which is synthesized on the leading strand, whereas the low molecular weight peak represents the fragments, the Okazaki fragments, all right, which are not able to replicate further. Why? Because the organism E. coli, it lacks the DNA ligase enzyme, and DNA ligase is, if it is absent, then the fragments won't be able to join together, okay, and then that this strand won't be able to participate in the next replication process. Okay, so this is clear. So this was the end of this session. I hope you have enjoyed it. Thank you very much.